So friends, in this video, I'm going to take this topic gluconeogenesis, and also in the same video, I will be helping you learn which amino acid is glucogenic, which is ketogenic, which is in both category. I'll make this very easy for you. So let's get started. Gluconeogenesis. And also before starting with this video, please see the video of seen in fasting state. What happens in fasting state? And you know that in fasting state, link reaction is inhibited because in fed state, insulin is there. Insulin will activate the link reaction enzyme pyruvate dehydrogenase complex, and pyruvate gets converted to acetyl-CoA, and in fed state this acetyl-CoA is used for the synthesis of fats in the body, right? But on the other hand, in case of fasting situation, you know there is beta oxidation of fatty acid occurring, and because of beta oxidation of fatty acid, there is lots of acetyl-CoA, which is the product of link reaction. So when there is lots of acetyl-CoA, the product of link reaction, acetyl-CoA, this excess acetyl-CoA will itself inhibit the enzyme pyruvate dehydrogenase complex. So now this pyruvate is not used up for the formation of acetyl-CoA. So this pyruvate will be used up for the formation of oxaloacetate. This oxaloacetate is 4-carbon compound. Pyruvate is 3-carbon compound. So from 3 to 4 carbon, we have to add carbon dioxide, and name of this enzyme is pyruvate carboxylase, which is the first step of gluconeogenesis. This is the first step of gluconeogenesis pathway, and this first step is also activated by this molecule acetyl-CoA. I told you that in fasting state, acetyl-CoA has 3 fates. Either it can go into TCA, or it can go into synthesis of ketone bodies pathway, or it can go into activation of gluconeogenesis. And in activation of gluconeogenesis step, this first step of gluconeogenesis is activated, right? Now, gluconeogenesis pathway occurs in which compartment? This pathway occurs in both mitochondria and cytoplasm, and any pathway which occurs both mitochondria and cytoplasm, that pathway will start from mitochondria. It will start from mitochondria. Now, let's talk about organ. It is occurring in which organ? It is occurring in liver and kidney. And as this gluconeogenesis pathway is occurring in fasting condition, so the enzymes will be active in phosphorylated state, and all these enzymes will be activated by which hormone? Activated by glucagon and inhibited by insulin, right? Now there is a rule of 2 for gluconeogenesis. Rule of 2 for gluconeogenesis, that is, it occurs in 2 compartments, occurs in 2 organs, and occurs in 2 situations. The 2 compartments you know are mitochondria and cytoplasm, 2 organs are liver and kidney, and 2 situations. One situation I'm taking, not eating food, so fasting or starvation, that is one situation. And second situation is diabetes. In diabetes, gluconeogenesis will be activated. See, in diabetes, there is lots of glucose, there is hyperglycemia. Still, body is making more glucose, gluconeogenesis occurring. Why? Because cells are not receiving glucose. The problem is that insulin is not there, so GLUT4 is not activated. So the channel, glucose transport channel GLUT4 on the peripheral cell of the body is not opened, right? So glucose cannot enter into the cells. So cells will think that we are in fasting, right? So cells will try to make glucose, so body will start gluconeogenesis pathway, right? Now, you know we have done glycolysis. Glycolysis is glucose to pyruvate formation, and this gluconeogenesis is opposite. Gluconeogenesis is opposite, that is pyruvate to glucose formation. So can I say that gluconeogenesis is just the opposite of glycolysis? No, this line is wrong. We cannot say that because glycolysis pathway is irreversible. We have 3 irreversible steps in glycolysis. Those 3 steps cannot be reversed. So therefore, we cannot say this line that gluconeogenesis is just the reversal. No, different enzymes will be used in gluconeogenesis instead of those irreversible enzymes of glycolysis. Let's see those enzymes. In glycolysis, there are three irreversible enzymes: pyruvate kinase, PFK1 and hexokinase. So these same enzymes cannot be used in gluconeogenesis. Instead of these enzymes, we're using some other enzymes in gluconeogenesis. That is, instead of pyruvate kinase, we have to use two enzymes, that is pyruvate carboxylase and PEPCK, full form phosphoenol pyruvate carboxykinase. And instead of PFK1, we are using fructose 1,6-bisphosphatase, and instead of hexokinase, we are using glucose 6-phosphatase. So now let's see the exact pathway. See the diagram. This is mitochondria shown here. This is cytoplasm shown here. Glycolysis is glucose to pyruvate occurring only in cytoplasm, but gluconeogenesis is occurring both in mitochondria, cytoplasm. It will start from mitochondria. So starting from pyruvate like this is the route of gluconeogenesis. First step in mitochondria and then the next steps in cytoplasm. So let's see the first step which is occurring in mitochondria. We have already done with that. That is pyruvate to oxaloacetate, 3 to 4 carbon. Enzyme is pyruvate carboxylase. This is the first enzyme of gluconeogenesis. And I already told you this enzyme is activated by acetyl-CoA. This activation of acetyl-CoA, this activation by acetyl-CoA is very, very important, right? Now, oxaloacetate is formed. Now, oxaloacetate has to come out in cytoplasm because other steps will occur in cytoplasm. But oxaloacetate cannot cross in a mitochondrial membrane. So here it will take the help of malate, that is oxaloacetate will form malate. Malate can cross in a mitochondrial membrane. In cytoplasm again oxaloacetate is formed. So we are taking the help of malate. Now tell me, is it malate shuttle? Is it malate shuttle? I'll give you one hint. The hint is that here NADH is used and here in cytoplasm NADH is produced. See, this is happening opposite to the purpose of malate shuttle. Malate shuttle's purpose was to take NADH from cytoplasm to mitochondria so that it can take part in ETC. But here too, opposite is happening, right? So this is not malate shuttle. Your guides have written this is malate shuttle, but that is wrong. It is not malate shuttle because NADH is coming outside in cytoplasm, right? See, here in this gluconeogenesis, we want to bring oxaloacetate outside. We don't care what is happening to NADH, right? Let anything happen to NADH here, but we want to bring oxaloacetate outside. That is the purpose here. So this is not malate shuttle. Now see the next step, that is oxaloacetate to phosphoenol pyruvate we have to see. You can see in cytoplasm when phosphoenol pyruvate to pyruvate was occurring, that was occurring with the help of enzyme pyruvate kinase. But this pyruvate kinase is irreversible. That's why now we have to go opposite, pyruvate to phosphoenol pyruvate. We are going like this. First step in mitochondria, then in cytoplasm. And these two enzymes are used, that is pyruvate carboxylase and the second one is phosphoenol pyruvate carboxykinase. So let's see the step, oxaloacetate to phosphoenol pyruvate. Oxaloacetate is 4 carbon, phosphoenol pyruvate is 3 carbon with a phosphate. So from 4 to 3 carbon, we have to remove carbon dioxide and we have to add a phosphate. For adding that phosphate, GTP is used. Earlier also I told you that thiokinase of TCA produces GTP in liver and kidney for gluconeogenesis, right, during starvation. So this step of gluconeogenesis, PEPCK enzyme requires GTP, right? And the EC number of PEPCK enzyme is 4, that is, it is belonging to lyase category. Now see the next steps, phosphoenol pyruvate to fructose 1,6-bisphosphate. There are so many steps of glycolysis which are occurring here. Like in opposite direction now we have to go. All these enzymes of glycolysis are reversible enzymes. So we can use the same enzymes of glycolysis here for in gluconeogenesis also because they are reversible, right? And in next step which was occurring in glycolysis, that is fructose 6 to fructose 1,6. Here the enzyme is PFK1 in glycolysis, but PFK1 is irreversible, so we have to use some other enzyme in the opposite direction. In the opposite direction, what we are doing is fructose 1,6-bisphosphate is getting converted to fructose 6-phosphate. You can see phosphate is getting removed from number 1 position here. So whenever phosphate gets removed, the name of enzyme is phosphatase. Full name of enzyme will be, take the name of substrate and add phosphatase. So it will be fructose 1,6-bisphosphatase, right? Now next step, fructose 6 to glucose 6 phosphate, that is, they are isomers of each other. The enzyme is isomerase. This is reversible. So the same enzyme you have seen in glycolysis also. So reversible enzyme, so that is same in glycolysis and gluconeogenesis. Now the last step here, in glycolysis, glucose to glucose 6 phosphate was done by hexokinase or glucokinase, right? Now instead of that, we have to use different enzyme in opposite direction because hexokinase again is irreversible. Now in the opposite direction, we have to do glucose 6 phosphate to glucose. So again, we have to remove phosphate here. So name of enzyme will be glucose 6 phosphatase, right? So we have 4 special enzymes in gluconeogenesis: pyruvate carboxylase, PepCK, fructose 1,6 bisphosphatase, and glucose 6 phosphatase. Let's see the compartment. Pyruvate carboxylase is very clear, is present in mitochondria. PepCK is present in cytoplasm. Fructose 1,6 bisphosphatase is present in cytoplasm. But this last one, glucose 6 phosphatase, is not in cytoplasm. We cannot keep this enzyme in cytoplasm because you know that whenever glucose enters a cell, quickly it is converted to glucose 6 phosphate. If this enzyme is present in cytoplasm, then that glucose 6 phosphate will be broken down by this enzyme. So we cannot keep this enzyme in cytoplasm. This enzyme is present in endoplasmic reticulum. Let's see the diagram. See this diagram. This is the membrane of endoplasmic reticulum. Membrane of endoplasmic reticulum. In the membrane of endoplasmic reticulum, here we have this enzyme glucose 6 phosphatase present. Now this is cytoplasm. In cytoplasm, when in fasting condition gluconeogenesis is occurring, then it can form glucose 6 phosphate. All the steps which are occurring in cytoplasm, they will occur till the formation of glucose 6 phosphate. Now breakage of glucose 6 phosphate cannot occur in cytoplasm. So this glucose 6 phosphate will enter into endoplasmic reticulum lumen. Here the reaction will occur. Note here that enzyme is present in the membrane and reaction is occurring in the lumen of endoplasmic reticulum. So glucose 6 phosphate is broken down to glucose and inorganic phosphate. They can again come out in the cytoplasm. That is glucose released in the cytoplasm, and this is mostly liver cell. Gluconeogenesis occurring mostly in liver cell. So via this GLUT2 it can come out, and this way liver is giving glucose to blood, right? So very beautifully this enzyme is hidden inside endoplasmic reticulum so that this last reaction of gluconeogenesis occurs in endoplasmic reticulum. So can I say that the sequence of compartment for gluconeogenesis is that first step in mitochondria, then next many steps in cytoplasm, and then the last step in endoplasmic reticulum. This is the sequence of compartments of gluconeogenesis, right? Also see one more thing in this diagram, that there is T1 transporter for the entry of glucose 6 phosphate in the endoplasmic reticulum. There is T2 transporter for the exit of glucose. There is T3 transporter for the exit of inorganic phosphate. Can I say all these transporters or channels will be active only during fasting? They will be active only during fasting because in fed state we want this glucose 6 phosphate to enter glycolysis, but in fasting state we want this to be broken down so that it forms glucose which can be given to blood, right? Now next we have to do the substrates of gluconeogenesis. Mostly 3 carbon compounds are good substrates of gluconeogenesis. You can see first 4 which I have written, they all are 3 carbon compounds. First is pyruvate. Pyruvate can form glucose, this you have seen. Now next is lactate. Lactate forms glucose by forming pyruvate, and then pyruvate can form glucose, right? So first pair is of pyruvate lactate. Next pair is of glycerol propionic acid. I already told you that fats can never be converted to carbohydrates, but there are 2 exceptions, that glycerol and propionic acid are 2 breakdown product of fats which can form glucose, right? Then next is amino acids. Next pair is of amino acid. You know that we have 3 types of amino acids: glucogenic category, both category, and ketogenic category. So which category can make glucose? Glucogenic category can make glucose and both category can make glucose. So you can learn it like this, that first pair is of pyruvate lactate, second pair is of glycerol and propionic acid, and third pair is of amino acids, right? So these 6 are substrates, plus any TCA intermediate is a very good substrate for gluconeogenesis. Any TCA intermediate. How? Because any TCA intermediate will form oxaloacetate in the end, and oxaloacetate can make glucose. We have seen in our pathway oxaloacetate is forming glucose, right? Now we have 3 types of amino acids: ketogenic category, glucogenic category, and both category. Let's learn these amino acids, which amino acid belonging to which category. I will make it very easy for you to learn. See here, amino acid, any amino acid which forms acetyl-CoA in the end, then it is ketogenic or glucogenic? Acetyl-CoA can form ketone body or glucose? I told you acetyl-CoA can only form ketone body. It is the starting material for ketone body. But acetyl-CoA is never glucogenic. It can never form glucose, right? So any amino acid which is forming acetyl-CoA as the end product, then that amino acid is ketogenic, right? Or if any amino acid is forming ketone body as the end product, then also it is ketogenic. Now see the examples. We have only two ketogenic amino acids, both starting from L. So very easy to learn. Leucine and lysine both are ketogenic. But see, lysine is in controversy. Lysine is in controversy. So you just need to know it is in controversy. Rest you can solve the questions. Don't keep wasting time on the controversy, because whenever question on some controversy is asked, the question is framed in that way that examiner also knows it is a controversy. So once you know it is a controversy, then you can solve the question. For example, now you know lysine is in controversy. So if I ask you a question, which is ketogenic, and both are given, leucine, lysine both are given, so which one will you mark? You will mark leucine, right? Because you know that lysine is in controversy, right? Okay. Now coming to glucogenic category, any amino acid which will form TCA intermediate is in the end, or any amino acid which will form pyruvate in the end. You know, any TCA intermediate can form glucose, pyruvate can also form glucose. So such an amino acid will be glucogenic, and there are many glucogenic amino acids. We will not cram all the glucogenic, but we will use a smart method of learning here, that we have already learned amino acids in ketogenic category. Now very few amino acids are present in both category, so we will learn them, and remaining all will be in glucogenic category, right? Now see both category. In both category we have a mnemonic tip, TIP, tip. From T I have written tryptophan, tyrosine, and I am writing threonine in front of you, because I want to tell you this thing, that threonine was earlier in controversy, but now it is clear in your standard books that threonine is in both category. So now it is very easy for you to learn. Any amino acid starting from T. There are only 3 amino acids starting from T. They all are in both category. So easy to learn, right? Next, isoleucine. I is isoleucine, the mnemonic tip. T is all the 3 amino acids starting from T. I is isoleucine. P is phenylalanine. But be careful, this is not proline. This is phenylalanine, right? So how many amino acids we have in both category? We have 5 amino acids in both category. And you know we have 2 amino acids in ketogenic category. So out of 20, 7 are done. So remaining 13 amino acids are glucogenic amino acids, in which most glucogenic amino acid is alanine. I told you Cahill cycle. So alanine is most glucogenic amino acid here. Now see one more thing. Purely ketogenic amino acids are 2. Purely glucogenic amino acids are 13. And both category contains 5. So tell me, in total, how many are ketogenic? In total, how many are ketogenic? 2 plus 5 is equal to 7. In total, 7 amino acids are ketogenic. In total, how many are glucogenic? 13 plus 5, that is 18 amino acids in total are glucogenic. Can you see? Out of 20, 18 amino acids can form glucose. Only 2 cannot form glucose, and they are leucine and lysine. They can never form glucose, right? Now let's see the energetics of gluconeogenesis. You know pyruvate mainly is converted to glucose. Now pyruvate is a 3-carbon compound and glucose is a 6-carbon compound. So we have to take 2 molecules of pyruvate to make 1 molecule of glucose, right? So everything will be multiplied by 2 in energetics. So when we are taking 1 pyruvate, then first enzyme used is carboxylase, and you know that any carboxylase will use ATP, right? Then the second enzyme uses phosphoenol pyruvate carboxykinase, which will use GTP. This enzyme will use GTP. Then I told you from phosphoenol pyruvate to fructose 1,6-bisphosphate, all the enzymes of glycolysis will be used. And here only one enzyme is there which is using ATP, and that is phosphoglycerate kinase. PG kinase, phosphoglycerate kinase will use ATP. When PG kinase was involved in glycolysis, it was producing ATP. Now in opposite direction, when PG kinase enzyme will work, it will be doing opposite. It will be using ATP, right? So 2 ATPs and 1 GTP is used, but everything will be multiplied by 2. So see here, pyruvate carboxylase is using ATP, PepCK is using GTP, PG kinase uses GTP. So this all is multiplied by 2. So it will be 4 ATP and 2 GTP, or you can say 6 high energy phosphates are used. 6 high energy phosphates are used to make glucose from 2 molecules of pyruvate, right? Also, if they ask you 2 molecules of lactate, also 6 ATPs will be used, because lactate will form pyruvate, and here there is no use or production of ATP in the conversion of lactate to pyruvate. Again, if they ask you one more thing, 2 alanine will make glucose, and here also same 6 ATPs will be used, because alanine can be converted to pyruvate, and in the conversion of alanine to pyruvate there is no use or production of ATP, right? So from pyruvate, lactate and alanine, all 3 are making glucose, and 2 molecules will be used to make glucose and 6 ATP.